Hello everybody and welcome to Bite Size Biology, and today we are continuing on with the plant videos, and specifically we're going to focus in on plant reproduction. So with plant reproduction there's quite a few things that can come up. You could be asked to draw a Lib diagram of the carpel and the seed. You could be asked to describe pollen formation, the egg and polar nuclei, fertilization processes, which there's quite a bit in number 2 there. State the process of pollination itself. Name state how seeds and fruit are dispersed. Very similar methods, however. Identify the benefits of dormancy to plants. State the factors necessary for germination. There is an experiment involved in this one. That's to show digestive activity of a seed. And you should also be able to describe the changes in dry mass of a seed throughout germination. And that's those graphs that you might be familiar with. So look, we'll go through them, and you'll see the marks are actually very generous for this. So even though there's quite a bit there, they're very generous marks. And if you have these learned off, like, you could almost be guaranteed to get full marks. So definitely one to worth looking at. And I know students, and my own students are including this, they like to avoid this chapter if possible. But like, it's definitely the better one to do. Compared to human reproduction, this is actually easier. Less interesting for sure, but easier to get full marks in. So 2020, and you might be familiar with some of the other videos at this stage, but 2020 asked an awful lot of plant questions. Almost every chapter in plants came up, which doesn't always happen. So 2020, answer the following in your knowledge of sex reproduction following plants, describe in detail the development of the pollen grain and the events that happen to mature. Okay, look, we'll start with one first. The pollen grain development, which we have here. Now, so how are pollen produced? This is the male gamete essentially. Okay, so all that happens there is meiosis occurs, producing a tetrad. There are 4 haploid cells. Okay, the tetrad breaks up, forming 4 pollen grains, and this all takes place in the anther of the pollen. So, you've got the stalk of the pollen, you've got the head of the pollen, which is the anther. Meiosis occurs there, producing a tetrad. That's 4 haploid cells. Each of those 4 haploid cells form a pollen grain. And that's it. Males are actually quite easy for plant reproduction. And again, the word meiosis is important here. Okay, anything that involves the formation of gametes, be it human or plants, is meiosis. Pretty much everything else then after that is going to be mitosis. Now, describe in detail the events that happen to a mature pollen grain following pollination up to double fertilization. Okay, what's actually happening here? Well, 3 points here. I just state that the pollen grain lands on the stigma and produces a tube nucleus. Now, a pollen grain consists of 2 things. One is the generative nucleus, which is here, and the other one is the tube nucleus. So the tube nucleus forms, and that basically goes down all the way down the stigma towards the through the carpel towards the ovule. Now, the generative nucleus falls down the tube nucleus, and it divides by mitosis. And again, the word mitosis is important here. Everything after the pollen being produced is mitosis. And it produces 2 sperm nuclei as it travels down the tube nucleus. And that's really important, because one of the sperm nuclei is going to fuse with the egg, and the other sperm nuclei is going to fuse with the 2 polar nuclei. And we call this double fertilization. So that's the process of fertilization here. We'll be talking about this later on as well, and specifically double fertilization. Give 2 adaptations of wind-pollinated flowers that increase their chance of successful pollination. So if they're wind-pollinated, they're often going to produce lots of pollen, okay, and the pollen produced is going to be very, very light, so that it can travel by the wind. Remember, plants want their seeds, their pollen, everything to be transported as far away from the plant as possible to reduce competition. So the pollen produced is very light, produces lots of pollen there. The anther is feathery, okay, and it's usually hanging outside of the plant, outside of the feather, outside of the flower. So there's a few different adaptations there, I said. And 2 advantages of sexual reproduction versus asexual reproduction. The common enough question comes up in another chapter as well called vegetative propagation, which we will look at next. So what's good about sexual reproduction? Greater survival chances, okay, because they're more likely to show adaptations, and evolution will take place over time, a longer period of time, of course. And you also have greater variation, and obviously greater variation leads to those survival chances increasing. Sticking with 2020, students were asked in that year about the experiment, and this is to investigate the digestive activity of seeds during germination. Now, part A, which is standard enough that they will ask for this, the theory part of the chapter. What is meant by the term germination? It is the regrowth of an embryo after a period of dormancy, providing that there are suitable conditions necessary. The market schemes haven't been released yet, although I think they are going to be released very, very soon for the 2020 ones. So I wasn't sure if suitable conditions was necessary or not, but I put it in. So the regrowth of an embryo after a period of dormancy, providing that there are suitable conditions. Suitable conditions being temperature being okay, and there being enough oxygen and so forth. Say 3 factors necessary for germination: temperature, oxygen, water. So, depending on which version you did on this, it might be slightly different. So you might have used starch agar or skimmed milk agar. I use starch agar. I think most people do use starch agar. I will only be referring to starch agar part of it. So that this does actually matter for the rest of it. So why were the seeds soaked in water prior to the investigation? 2 reasons: to soften the testa, that's the coat of the seed, and also to activate the enzymes. Part 2. Describe how you further prepared the seeds and added them to the agar plates. Now again, there's no mark scheme available just yet for this, but generally what you do is you cut the seeds in half and you sterilise. You sterilise it so that no microorganisms can interfere with your result. You then use a tweezers and you place the open side face down on the starch agar, and you leave it there for approximately 5 days. That should give you full marks for that. Under what conditions did you store the plates containing the seeds to allow germination to occur? Well, you made sure that you had the correct temperature. You don't need to specify what temperature, just that it was the correct temperature. How did you show that digestion had occurred? Well, that's the last part. You basically flood the agar plates with iodine, and you'll notice the colour change, if there is any colour change. And the result you would see is it would go blue-black, except the entire plate would go blue-black, except for underneath where the seed was, because starch is no longer present there. What's happened there is the amylase inside the seed digested all the starch, so now you only have maltose there, so therefore it remains brown. So the plate goes blue-black except for underneath the seeds, and it remains brown there. 2019. Describe in detail the development of the embryo sac from a megaspore mother cell. Okay. So basically what they want to do is, how was the egg and how was the polar nuclei formed? So this is for the female version of it now. So the megaspore divides by meiosis to form 4 haploid cells, which is standard enough. Meiosis always produces 4 haploid cells. Now, out of those 4 haploid cells, 3 of them will die. So you're left with 1 haploid cell. And what happens then is that haploid cell is the embryo sac now. So that's what we call it. That will divide by mitosis 3 times, and it forms 8 haploid nuclei. Remember, if it divides by mitosis, it's maintaining the number of chromosomes produced. So 8 haploid nuclei. And you can do that out and you'll get 8. Now, out of the 8 haploid nuclei, 5 will die. 2 are going to be classified as the polar nuclei, and 1 as the egg. So there are my 3 steps there that are involved in the development of the embryo sac. So it's really, really important that you do know this, because it does come up every now and then. Okay, next. double fertilization, what happens? So I said earlier on that I would go back to this. So I just write it out like this, nice and simple. The sperm nucleus, you could just say the sperm cell, it doesn't matter, and the egg fuses to form the zygote. So similar to humans, a zygote is formed. Now it's double fertilization, so you also get sperm nucleus plus 2 polar nuclei fuse, and they form an endosperm. So the endosperm here, and the endosperm is unique in which it is triploid. Now you didn't need to say this, but just to put that in, so it is triploid, which means it's 3N. Again, that wasn't necessary, but just a little bit of extra detail, that was all, just for you guys. For part 3, draw a large label diagram of a seed, okay, and put in testa, pumule, and radicle. Okay, so look, this is my diagram here. You can see it's pretty bad. It doesn't need to be good. You just need to have it good enough that you can label it. So the pumule is the one that's going on top. The radicle is going to be the roots. So the pumule is the shoot. The radicle is the going to form the roots. The testa is the coat. That's it. Don't overcomplicate it. It doesn't need to be, and you do not need to be colouring that in or anything like that. 2018, define dispersal in relation to seeds and 2 reasons why dispersal is important to plants. Well, there's loads of different things. First off, dispersal of seeds is the carrying of seeds away from the parent plant. And why do plants want that? Well, it reduces competition. They're not going to be competing for as much light or water if the plants are spread out apart as much as possible. To avoid overcrowding, so overcrowding obviously runs into the similar problems as competition, but also you could increase numbers in new areas so that it ensures the continuation of that species of plant. So dispersal is very important. Give one way in which the knowledge of seed dormancy is useful to humans. This question comes up quite often. So why do we need to know seed dormancy? Well, we need to know it to maximise the growing seasons, and also to have the correct temperatures for storage. Really, really important that you know this one. Now, I have 2 reasons here, okay? And the reason I put in 2 is because quite often you're asked for 2 reasons, not 1. So just be careful with that. So, to maximise the growing season and to ensure that we have the correct temperatures for storage. That's why we need to know about seed dormancy. Other than water, name 2 essential environmental requirements for the successful germination of seeds. Now you only have to name them, you didn't have to state what they do. So that's going to be oxygen and temperature. Now, I don't want you saying just temperature. I want you saying a suitable temperature. Okay, so that's important there, okay? It's a subtle change. Okay, temperature could be anything. It could be unsuitable. It could be suitable. It's too vague. Okay, and there's no point leaving it up to chance for your leaving cert. So oxygen and a suitable temperature. Temperature comes back to, again, it goes back to enzymes. Anything with temperature goes back to enzymes. Oxygen's got to do with respiration. What is the role of digestion and the role of respiration in seed germination? Well, why do we need digestion? You just need to, and it's the same thing always, is to provide soluble nutrients. So digestion, to provide soluble nutrients. Respiration, you just say what it is. It's a release of energy for growth. Okay? It didn't say aerobic or anaerobic, so you just leave it as the release of energy for growth. Now, you're given a graph here, everybody, and this is going to be common enough. So there's 2 types of graph you're going to see. So this graph here has dry mass on the y-axis and time on the x-axis, common enough. And you can see there's an initial decrease, and then there's an increase, and then we go back to a decrease again. So you're asked in the graph to give a reason for the initial decrease in mass. So this is the dry mass of peas. It doesn't matter what it is, to be honest. Okay. So why is the dry mass of seeds decreasing? Well, it's decreasing because food is being used up. Respiration is occurring. Okay, so this is decreasing in mass here because food is being used up. Respiration is occurring. Okay. And then the subsequent increase in mass. Well, in this case here, it's increasing in mass due to photosynthesis. More than likely around week 1, sometime around week 1 here, at the end of it, okay, the plant had produced leaves, and leaves obviously incur photosynthesis, so therefore the mass increased because food was being produced. Name a substance which, because of its change in quantities in seeds, could be responsible for the changes shown in the graph. Okay, that always comes back to a few different things, but you could say glucose, starch, or lipids. So glucose, starch, or lipids. Okay. 2017. I really like this question. Now, if you haven't done sexual reproduction for humans, haven't studied that chapter yet, well then this mightn't be a great question for you, okay? Just to bear that in mind, because it compares the two of them. So, what type of cell division occurs? Production of female gametes in flowering plants and in humans. If more than one type occurs, in either case, write them in the order in which they occur. So, flowering plants, well, we said it earlier, meiosis and then mitosis. In humans, it's just meiosis. Because mitosis must occur 3 times for the egg and the polar nuclei to be formed. Precise location of the female gametes in flowering plants, okay, it's the embryo sac or the ovule. Ovule is perfectly fine to say. Embryo sac, grand. In humans, okay, you can say the ovary, or you can be specific about it, and that's the graphene follicle. Give a location of fertilization. Okay, so in plants, it takes place in the ovule. Again, the embryo sac. Doesn't matter. And in humans, it's the fallopian tube. Name the next stage of development following the formation of a zygote. So you have your zygote, and the next step for flowering plants is the embryo. Okay, and that's really as far as you get for plants. Embryo and then the plant. Okay, for humans though, it's a bit more complicated. You've got the zygote, you've got the morula, you've got the blastocyst, and you've got your fetus and baby and so forth. Okay. An advantage and disadvantage of sexual reproduction. An advantage is increased variation, greater chances of survival, and there's loads you could say for us. A disadvantage though is it's slow because it requires 2 parents. So if it's asexual reproduction, it's going to be much quicker because only 1 parent is necessary. You could also refer to a disadvantage being increased variation. So for some people in agriculture, they want to make sure that the plants they have is top quality, and they want to are as high quality as possible, and therefore variation might actually be problematic for them. So it could be a disadvantage. So the diagram here shows details of sexual reproduction in a flowering plant. Named parts A, B, and C. So A is up here, so that's the stigma, the sticky stigma, which captures the pollen, which is obviously D. B is this part here, that's the pollen tube, in which the generative nucleus will come down here and it will divide by mitosis to form 2 sperm nucleus, which is E. And then C. Well, C looks like an egg, doesn't it? A fried egg, and that's what it is. It's on its own. These 2 over here are the polar nuclei. F there is the ovule there, or the ovary, I should say, and then the inside of it is the ovule. So just 2 ways in which structure D could have arrived at its position. So D is the pollen. So how could it have arrived there? There's a couple of different things. You could say wind, insect, animals. Insect and animals are the same thing. By what process does the structure labeled D give rise to structure labeled E? Well, if it's always going from 1 to 2, it has to be mitosis. If it goes from 1 to 4, it's meiosis. So in this case here, mitosis. Give the fate of each of those. I've said it to you guys already. 1 sperm nucleus will fuse with the egg to form the zygote. 1 sperm nucleus will fuse with the 2 polar nuclei to form the endosperm. Endosperm is like the food for a seed. Name the structure labelled F and say what it develops into after fertilization. So F there is the ovule here. Okay, so this overall is the ovary and F is the ovule, and this is your embryo sac, which is the middle part here at the center. So F is the ovule, and what's the question there? It develops into the seed. So there's 2 parts there, the ovule and the fruit. The ovule and the ovary, what do they develop into? The ovule is the seed, the ovary is the fruit. An easy way to remember it is ovary has an ore in it, and so does fruit. Okay, you weren't obviously asked for it here, but you are asked for it later on. So ovule forms into seed, which makes sense because that has to be somewhat in the inside the ovary, because fruit often surrounds the seed. If you think of any apples or whatever, okay, the seed is on the inside. Okay, so I said before that you will get two types of graph, and this is the second type of graph you're going to see. Okay, now we'll get to that now in a moment. Before we get to that, let's see what we have over here. Following fertilization, from precisely which part of the flower is the seed formed? Oh yeah, so which part of, now really that should be plant more than anything, but anyhow, seed. So again, seed is the ovule, which we have over here. What is meant by the term monocotyledous? So that's one seed leaf. That's referring specifically back to the plant structure chapter, in which I have a video done already for you guys. Name two biomolecules stored in the endosperm or the cotyledon tissue. Well, there's a few there. You could say your lipids, you could say your proteins, you could say your carbohydrates. Any of those three would be fine. Lipids, carbohydrates, proteins. Give one way in which knowledge of seed dormancy is useful to humans. Well, we said that already. That's to maximize the growing season, and I didn't put it in there, but to know the correct temperatures for seeds during dormancy. V. This is a graph here. Okay, y-axis is dry mass, the x-axis is time. So that's standard enough. That's always been the same. And you can see that the embryo is increasing in mass over time, and the endosperm is decreasing in mass over time. Okay, suggests why the measurement of dry mass is preferred in these investigations. So why do they use dry mass, and why don't they just say the mass of the seed? Well, the problem here is if you use the mass of the seed, you're also including the water weight, the water mass in that, which is actually a problem because the water mass will be different for different seeds, even the same type of seed. It just depends if one seed got more water than the other one. So it's important to make sure that we get rid of any water in it. So what did I say here? So water content will vary among seeds. If you want this to be a fair investigation, okay, you need to make sure the water is gone. Explain why the mass of endosperm tissue decreases over the 3 weeks. So why is it decreasing here? Well, it's been used up. And why is it being used up? Respiration, which we have here. Part 3. What process begins at A? So you can see the endosperm, as that's decreasing, the embryo is getting bigger, which makes sense. But then, once we get to a certain point, the embryo shoots up in dry mass. What is happening there? Photosynthesis, which we have over here. And then, part 4. Would you expect the total dry mass of the seed to have remained the same up to A? And explain your answer. So would you expect the embryo plus endosperm dry mass to remain the same up to A, and to explain your answer? Well, I said no, okay, because it will lose mass in terms of respiration. Yeah, that was probably the best way of putting it, to be honest. Okay, I can't really think of anything else to say for that. That the mass would actually not be the same because respiration. Remember, respiration produces heat, and that energy is lost, it's dissipated into the soil and so forth. Okay, it's only tiny amounts, but it's still present. So, would it remain the same overall? I said no, because it would be lost due to respiration. 2014, everybody. Name the site of the production of a pollen grain. So that's the anther, as we said earlier. And the structure it must land on to complete pollination. So it goes from the anther to the stigma. Name two methods of cross-pollination. Okay, so that's insect, wind. Many species of plant have mechanisms that prevent self-pollination. That means they're pollinating with themselves. Suggest how plants could benefit from this. Okay, well, if you have cross-pollination taking place instead of self-pollination, what's going to happen there is you're going to get greater variation occurring, which is a good thing. Describe in detail the events that follow the pollen grain at the destination referred to in part I, up to including fertilization. So look, this is what we have here. Pollen grain lands in the stigma, produces tube nucleus. Generative nucleus divides by mitosis, produces 2 sperm nuclei. The nuclei travel down the tube nucleus. One sperm nuclei will fuse with the egg to form the zygote. Another sperm nuclei fuses with the 2 polar nuclei to form the endosperm, double fertilization. You can just refer to it as double fertilization. And what part of the flower develops into the fruit? So fruit contains an ore, so think of fruit, ore for ovary. Okay, we're nearly there. Brief account of the following. We've answered this question before in a different video. What does the petal do? It attracts insects. What does the anther do? Produces pollen. And what does the stigma do? It captures pollen. It's often sticky to capture the pollen. Name one structure through which the pollen tube grows in order to reach the embryo sac. Well, that's the style or the ovary. Within the pollen tube, the generative nucleus divides to form 2 male gametes. What type of division takes place? Mitosis. With what does each male gamete fuse in the embryo sac? Well, we said this already, the egg and the polar nuclei, and named the product of them, the zygote and the endosperm. As the seed forms following fertilization, a food store develops in one of the 2 structures. Name any one of these 2 structures. You could say the cotyledon or the endosperm. Either or. They're both food stores. Now we're almost done. Second last question, I think. So, sepals, anther and stigma. Sepals, they just protect the flower. Okay, the anther produces pollen, the stigma captures the pollen. Okay, we've seen that come up before. Describe the development of pollen grains from the microspore mother cells. What's meant by fertilization, and give an account of fertilization in flowering plants. 27 marks. And we've done these all before. And you have to just learn them off. I know it's not easy. Megaspore, look, I'm not even going to call it out here. They're in the notes. You can just read that up in your own time. But this has to be known. What is meant by the term dormancy, and how is a dormancy of a benefit to a plant? Now, dormancy is a period of reduced metabolism, or you can say little or no metabolism too. But how is it useful for plants? Okay, we've talked about how knowing about it is useful for in agriculture, but why specifically plants? Okay, well, there's a better chance of survival. If the plant is dormant during winter and then it starts to germinate during springtime, the warmer temperatures means it's more likely to survive. It's just one way in which dormancy is useful to farmers and gardeners. We know that already. In each of these factors, describe the effect of the process of germination. So water, oxygen, suitable temperature. So what do? I like that question. Hasn't been asked in a while. Okay, so water softens the testa, activates enzymes. A suitable temperature, okay, enzyme activity, optimum enzyme activity. And then oxygen, okay, respiration. Needed for respiration. 1 and 2 is the radical and plumule, so that's probably going to be what does the root form from and what does the shoot form from. So or for root, or for radical, and the shoot is the plumule then. Okay. And that was it, guys. Yeah, that was it. 2010 was the last one I did. So a lot of repetition in that. Okay, and I know it's tricky enough to learn off, like the development of the pollen grains, the egg, the polar nuclei, and fertilization, all that. But it's very repetitive and definitely well worth practicing and going over. Okay. So yeah, look, I'm going to leave you at that, guys, and the best of luck with your studies.